Welcome to our second set of lessons in the DNA and proteins unit. So our lesson or lesson set of lessons this week is actually brought to you by the second cat in the series of cats that belong to my house, Trevor. So Trevor was originally T-Dog as his first nickname. So I would like to think of him as the original T-Dog before students started calling me T-Dog at school. But my wife also calls him Bean Boy or the boy who lives. So Bean Boy referring to his little toe beans. She thinks they are the cutest out of all of our cats. And the boy who lived being a little cheeky reference to Harry Potter. I guess because Trevor lost an eye at such a young age, I think she maybe thought without us sort of fostering him, he would have had no hope of adoption. So yeah, we're also privileged to have Trevor featuring in today's video as well, sleeping next to me. So hopefully he might wake up throughout the video and then be able to say hello to the camera. Last week, we had a look at the key concepts or key underpinning concepts in DNA. We talked about where DNA is found in these two different types of cells, so prokaryotic cells, things like bacteria, and eukaryotic cells, so us and all other multicellular organisms. We also talked about this idea that DNA is wrapped around histones in eukaryotic cells. And depending, when we zoom out even further, depending on what sort of view angle we're looking at it from, we can then see that that DNA wrapped around the histones forms a new structure called chromatin. And when we go through replication processes, segments of chromatin all organize themselves accordingly into what we call chromosomes. If you do have any issues with any of the content that was presented last week, as always, email me, schedule some time to catch up. I would be more than happy to go through it as long as it takes, just so we can sort of nail down. Because in reality, we are learning so many new terms, and so it's absolutely reasonable that the first, second, third, and 17th time that you hear a term like chromatin, you still may or may not know what it is. So yeah, just keep asking questions and I will be more than happy to support. Our final student understanding of last week looked at DNA being found in these, or connected to histones and found in linear chromosomes inside of eukaryotic organisms and bound in the nucleus. And so the first student understanding for this week is now looking at DNA in prokaryotic organisms, and we say that it is unbound and circular in prokaryotic organisms. So, unbound being not connected to a particular sort of key organelle, but also it's not protected by any cellular membrane as well, in that sort of same way that DNA within the nucleus is protected. We also know through a lot of extensive research that we have additional DNA, especially in eukaryotic organisms, called mitochondrial DNA. So found in a structure called the mitochondria within our cells. And also in plant cells, there is chloroplast DNA, and they support the functions of what those organelles are useful for. And we'll talk about that a little bit later. And there will be a table for that 1.D.I that I recommend you snipping tool out and put into your notes so that you can get that sort of full comparison of prokaryotic and eukaryotic chromosomes for that last student understanding. So we know that DNA, when we're getting ready to replicate, organizes itself into a structure called a chromosome. So for bacteria and for other prokaryotic organisms, chromosomes, they only contain a single chromosome. So very simple. But for us and for other complex organisms, there are multiple chromosomes as well. We don't get to see what those chromosomes look like until replication processes are occurring. We often see a spaghetti soup otherwise, and that was one of those pictures last week as well. But within the chromosome, we see the chromatin, but we also know that there's another term that we use when we look at segments of chromosomes called genes. And genes are made up of a series or a sequence of DNA that pretty much codes for the creation of something. So that could be a protein or an enzyme or something else. So it's kind of the instruction manual. Little bit of an additional set of recap. I won't spend too long on here. I'll actually even allow you just to pause this slide yourself. There is a pencil on the screen. Again, we've talked about this kind of stuff, so you don't actually need to renote it if you've already got it there. But feel free to pause that slide there and continue to take more notes as well if that's going to increase the depth of your understanding. For clarity purposes, this picture was on last week's set of slides, and probably may have caused a bit of confusion because it's more anatomically correct, essentially. It's actually got real life pictures of cells, and it's sort of got actual images of the substructures within the nucleus and the sort of the chromatin and the histones and things like that. So I guess probably the key things to just note is that if we were to look at a eukaryotic cell, you could see that big structure there, the nucleus. That's where we're going to find all of that DNA. That's where we're going to find all those histones. And then when we have a look at what they actually form, the DNA in this second part of this image, and let me get my pen here. In this image here, I don't know why it's doing that, but that there, when it's sort of getting ready to replicate, we can see that the DNA is wrapped very tightly around those histones, but then those chromatin fibers, which is the bulk of all of that, starts to get organized. So we talk about it as chromatin, and then we talk about it as chromosomes when it all gets organized, ready for replication. All right. And in the bacteria cell, we don't have that same sort of protection. We've got a single loop of DNA, and it is bound. No, it's not bound by anything. It's not enclosed by any kind of membrane. We call it a nucleoid region, and it's found within basically the main body of that cell called the cytosol. One of the questions you would have seen on Style last week would have been that we do contain DNA elsewhere in eukaryotic cells. And the first things that we're going to have a look at is the mitochondria. So the mitochondria is an organelle that is found in all eukaryotic cells. Its function is basically, we call it the powerhouse of the cell. And basically that's where the breakdown of sugar and the release of energy occurs to help power our bodies. And so it's reasonable to assume that the DNA that is found within that actually serves a purpose to help facilitate that occurring. So it does help with that sort of cell metabolism. It's also got some functions of apoptosis or adoptosis, I think it's called. I'm just going to look it up here. Apoptosis, yep, so cell death. So that can actually instigate a programmed cell death. That can occur in case we have mutations that we do not want during replication. So that can help sort of ensure overall survival of the organism so that cells that are dysfunctional do not actually go on to continue to replicate. And also the DNA as well within the mitochondria is said to have a role in oxidative stress control as well. You can look into those sort of things if you would like. But one thing to note is that the mitochondrial DNA is circular, all right, and it's not as specialized as the nuclear DNA. It's not the chromosomal DNA that we need for every single function. Chloroplasts within plants, so another substructure of plants or organelle within plants. Chloroplasts are where photosynthesis actually occurs. So that's that conversion of inorganic material into organic matter using sunlight. And so chloroplasts play a vital role in that. And there's DNA, a circular loop of DNA within the chloroplast that helps facilitate that actual function as well. So the sort of idea that we might touch on in the cells unit is that the theory is that all cells originated from a single point, so from simple celled organisms. And there was this point in time where this one sort of slightly bigger cell engulfed a mitochondrial cell, and they were both able to still continue to function. And so when replication occurred, the new cell the bigger cell had its own individual sort of all of the stuff that it needed, but also it had replicated the mitochondrial cell as well. And so over billions and billions of years, we get to this point where we get a lot more complex structures because the mitochondrial cell within the larger cell was able to perform functions such as breakdown of sugar for energy, which actually benefited the other cells, so it became more successful. Similarly, with the chloroplast, the theory is that that was a engulfing of a chloroplast cell type, a very simple celled organism, and then both the larger cell that had engulfed the chloroplast and the chloroplast itself were able to survive and keep doing their jobs. And then it was sort of what we call a symbiotic relationship where both were able to be successful. We'll talk about that in the cells unit though, so you don't have to know too much about it. You just have to understand that we do have additional DNA. It's not our chromosomal DNA, but it still serves a pretty vital role for the type of cells that it's in, so a plant or in all other eukaryotic cells. Like I said, the table on this particular slide, well worth you snipping tooling it out and chucking that in your notes. But if you're going to do so, make sure that you read and understand it. So quiz yourself on it as well, because like I said, no information is worth copying unless you actually understand it. So make sure you get it down, but also it makes sense to you. We now move into the next part of this, which is the replication processes that we're going to have a look at. So, replication of DNA, a key process that occurs, and that's basically the function of DNA, is just to keep replicating certain segments of it or the whole thing, so that we can actually do life processes, continue to grow and survive. And during these replication processes, genetic information is inherited. All right. DNA is doubled prior to cell division. Um, this concept is very, very key. This idea that we need to make a second copy of DNA before we actually create a new cell. Because if we want to produce two cells from one cell, we need to copy all the DNA so that we can then allow the new cell to have that identical copy of the DNA as well. If we didn't do that, the two cells would contain 50% of the original DNA that was in the first cell. So what happens is we get a doubling of DNA prior to cell division. And then that way we've sort of got, before the cell actually divides, we've essentially got 200% of what the DNA is. And then we can split it off so that both have the identical amount of DNA. And that's it. This ensures that both of those daughter cells, so the two new cells that are going to be created, have 100% of the DNA. What's really cool about that is that ensures that every cell arises from a pre-existing cell, is able to conduct all of the same processes that the pre-existing cell was able to do. And even though every single cell in our body contains all of the DNA, it doesn't actually mean that every single cell does the same function, but we'll talk about that a little bit later in this sort of unit of work. But there's a couple of prompting questions to sort of have a bit of a think about. Why might we need to replicate cells? Why is it important to have them contain the same information? And if all replicated cells contains the same information, how do some of them look and function in different ways? My group talked about that a little bit last year in terms of this concept of gene expression, but if you haven't, don't worry, we will cover that again. All right, when we compare prokaryotic and eukaryotic replication, so cells dividing, one cell becoming two cells, they do so via slightly different processes. So in prokaryotic replication, we call that binary fission, and it's quite a simple process. The end goal, though, between both of them is still the same. What happens is both of them do replicate DNA in both multicellular and unicellular organisms. Both of them actually go through a division process, and both of them in both of the brand new cells will have the exact copy of information. Okay. What you might notice on the eukaryotic replication image there is there's a few new terms. We will cover them in more depth, but feel free to chuck them into your glossary because they are tier 3 vocabulary as well. And some of those things like sister chromatid Does sound quite similar to chromatin, so I guess it's probably good to start noting that now so that we can start to get our heads around it through some repeated conversations about it.